Thank you for taking the time to listen to my talk. It is called Leptospira is essential for osmoadaptation in Leptospira biflexa. The work was done by myself, Herrera, and Dr. Christopher Pappas. My research is involving Leptospira, which are spirochetes found in the environment, and infect mammalian hosts such as rodents. They can be transferred to a water source or soil through the host's urine and either transferred back to another animal or potentially human beings. If transferred to a human being, it can cause diseases such as Wales disease and may lead to death. There's an estimated 1 million infections worldwide and 60,000 are fatal. Leptospira can remain motile in osmotic environments and use the osmotic change as an advantage for mammalian phase adaptation. From this idea, we generated a random transposon mutant library. My mentor inserted HEMAR1 into Leptospira, which created random transposon mutants. This means that it randomly inserted itself into the genome and disrupted random genes. My mentor gave me 4 mutants to study to see if they were sensitive to salt. I studied mutants 9.2, 9.27, 9.34, and 9.36. In order to study their sensitivity to salt, growth curves were set up for the mutants and the wild type. There were 2 technical replicates of each mutant and wild type and were grown in EMJH media, which is the standard way Leptospira is grown. They were grown under 17 millimolar salt concentration, which is a standard amount of salt where there isn't any osmotic stress on the cells, and 80 millimolar where there is osmotic pressure. The wild types were tested to ensure they grew well under each osmotic condition. Although what's being measured isn't cell counts but optical density, it still demonstrates a nice curve showing the bacteria's ability to grow. The wild type grew in the 17 millimolar salt concentration, but not in the 80 millimolar, and there was a significant difference between the wild types in these concentrations. All the mutants grew similarly to the wild type in the 17 millimolar salt concentration, and there was no significant differences between the mutants and the wild type. In the 80 millimolar salt concentration, mutant 9.2 grew similarly to the wild type and demonstrated no significant difference. Mutants 9.27, 9.34, and 9.36 didn't grow similarly to the wild type, and there were significant differences. Mutant 9.27 was a good finding since it showed no growth in the 80 millimolar salt concentration. This gene is gene Lepbia0192 and is responsible for NADPH quinone reductase. It codes for energy production and conversion in the cell. There hasn't been much research done on NADPH quinone reductase, but one study showed it plays a crucial role in maintaining oxidative stress in H. pylori, since it acts as an electron donor. There may be other genes involved in osmoadaptation in Leptospira, and future works could involve RNA sequence analysis on the transposon mutant library or testing the bacteria under different conditions to induce stress. I would like to thank the biology department for providing the resources for this research and Herrera for her collaboration. I would like to thank my mentor, Dr. Pappas, for his support and mentorship throughout this study.